Everyone, it's Miss Mansell. So we're going to cover actually both homogeneity and independence tests for chi-squared today. And I've tried to condense the notes down just to the nitty-gritty here. So reminding you that chi-squared test is a test for looking at what we call mismatch. We're trying to see if these distributions are the same or there's a relationship between an entire distribution of categorical variable. So we're looking at the mismatch between categorical variables or populations. All right, so last time you learned about goodness of fit test where you have a single sample, one categorical variable. And this is really how you can really distinguish between which kind of chi-square test. It's really about how the data was gathered. And so sometimes, as you're going to see a little bit later, you can use some words that can be misleading, and you just have to be really careful about the way that the data was collected. If you do the reading in the book tonight, it talks about that too. Just be really careful there. Okay, so just as a little summary here, I've included all the parts to the three different types of chi-square test. You have the Goff test or the goodness of fit test, one sample, one categorical variable. We're trying to see if a population has a certain hypothesized distribution and if this population distribution is the same as what's hypothesized. You've got your conditions, random, 10% if sampling without replacement, and our large counts are expected counts. And remember, expected counts are just calculated as n times p. So if you have, 84 tobacco plants and, 50% of them are expected to be yellow, then how many would you expect out of 84? And then the degrees of freedom is the number of subcategories. So you learned last time about the shape of the chi-square distribution and how you need to use the degrees of freedom in order to figure out what the p-value is, just like a t distribution. Okay, so the second type of chi-square test is called we can call it the HOP test, homogeneity of populations. Okay, so homogeneity means sameness. So in this case, we've got two or more samples from independent populations, and we have just one categorical variable that we're trying to look at, and the data is usually organized in a two-way table as opposed to goodness of fit, which is a one-way table. So what we're trying to do here is we're trying to see if homogeneity means sameness. So we want to see if the distribution of the categorical variable is the same for all populations, all of the separate independent populations. So the random sample condition, it's the same, except that it applies to multiple samples. If you're sampling without replacement, you would need to check all sample sizes to be the appropriate size. And what's different here is how you calculate expected counts. So expected counts are calculated as the row total divided by the column total over the table total. And we'll do an example in just a second. The degrees of freedom also changes because you're dealing with a two-way table. And so the degrees of freedom is the number of rows minus 1 times the number of columns minus 1. Okay, now these rows and columns, they are not including the headers, and they're not including the totals. So these are not including totals. So it's only the rows and columns of the actual data itself. Okay, so then what you do for the null and the alternative hypothesis here, remember that the null hypothesis is always a statement of no difference. And so we want to say that there is no difference in the distribution of whatever the categorical variable is. So this is going to change for the populations of whatever they happen to be. So you need your context here. So another way to say this is that the distributions are the same. You could say the distributions of the categorical variable are the same for all populations. The alternative, there is a difference in these distributions, aka the distributions are not the same. And as long as you have really good context in the null hypothesis, you don't need context in the alternative hypothesis. So then the calculation of the chi-squared test statistic and looking up degrees of freedom and the p-value and your conclusion is all the same as what you are familiar with. So lastly, we have independence and association. So again, this comes down to how was the data gathered? So this is kind of a combination of the other two. It's a single sample, but you have multiple categorical variables. And here, you're trying to see if there's a relationship between your categorical variables. Your random sampling condition and 10% condition are the same as they are for goodness of fit because it's a single sample. And then your large counts condition, because you're going to have a two-way table again, this is the same as the hop test. So where you take your expected counts, row total, column total, and divided by the table total. Your null and alternative hypothesis, you would say, remember there's, in the null hypothesis, there's no, here it's not difference between, it's that they're not related. So I'm going to say, not there, but the two variables, if they're not related, independent. So independence means no association. And in the alternative, the two variables are not independent. Or you could say are associated. I guess you could say dependent, if you prefer. So the thing about independence and homogeneity is that when you look at the two-way table is going to look the same in either case. And so you have to be sure to really pay attention to how the data was gathered. And that's going to tell you how, which test to use, not necessarily, what am I saying? You have to look at how the data were gathered. I'll just stop there because I'm going to move on. We'll look at some examples. Okay, whoopsie. All right, so here we go. Here's our finger length problem here. All right, so it says, is your index finger longer than your ring finger? Okay, so your index finger is this one and your ring finger is this one. Don't mind the manicure here. So does this depend on your gender? A random sample of 460 high school students in the US selected and asked to record if their point of blah, 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 blah. So here you've got your two-way table, and I want to just kind of highlight that these are the actual values. So when I do my expected counts, I'm just going to focus on those values that are there. So first, you're asked to make a graph to investigate the relationship between gender and finger length. So the appropriate graph for categorical data is a bar graph. You can do it stacked or you can do it side by side. And so I've got index finger, same finger, and ring finger. And because my sample sizes are not the same, I'm going to use proportions over here. So I'm going to go by 0.10. Okay, so then I'm also going to have a key. So I'm going to have female, and we'll use pink for male. And so then I need to think of or come up with the proportions. So out of the index fingers, let's see, 85 out of 158 is 0.54 roughly. And so I'm going to go up here and I'll write the proportions here at 0.54. And then for the males, they have to add up to 1. So this is going to be 0.46. For the same, 42 out of 86 is 0.49 if I round. 0.49. And so then that means that the males are 0.51. Oops, should make them the same width. For the ring finger, 100 out of 216 is 0.46. And that means that we've got 0.54. All right, so then we want to describe what we see. So what we see here is that, females tend to have a longer index finger compared to males. And for the other two fingers, males were higher. So you just write that, higher proportion of females had longer index fingers compared to males. While a higher proportion of males had ring fingers and I'm going to say ring fingers longer or ring and index fingers same length. Okay, so a little bit of a review from chapter 1. Okay, so then do the data provide convincing evidence of an alpha equals 0.05 level of an association between gender and relative finger length for your high school in the US? So again, remember that it's about, even though it says an association, we want to be careful here and make sure that we check how the data was gathered. So we have to ask ourselves, is it a single sample? Is it a single sample or is it multiple samples? So here it says it was a random sample. And so we have a random sample that was taken, and then each individual was categorized into several categories. One was gender, and the other was finger length. So the two categorical variables are gender and finger length. Because we have a single sample, then that means we're going to use the test for independence. Now it does say association, so maybe I want to make sure to use the word association in my null hypothesis. So I'm going to say, okay, so no association. There is no association between gender and finger length in the population of US high school students who filled out the Census at School survey. This is from the Census at School. I don't think it says that here. I got this out of our book, so um, you know, you could just cross this out if you didn't feel confident with that information. And so then the alternative is that there is an association. Okay, so we are told we have a random sample. Of US high school students. And we're most likely sampling without replacement, so let's check. Most likely sampling without replacement. However, is it reasonable that we have at least 4,600 high school students in the US? Sure. Um, uh, let's see, 4,600. What am I doing? I'm tired, it's late. There are at least 4,600 high school students in the population, so this condition is met. Okay, so then the expected counts. So we want all the expected counts to be at least 5. So for the first cell, we have 85. So, but remember the expected counts for, here, we'll come over here for a minute. So if I want the expected count for this cell, I'm going to take the row total, which is 158, times the column total, which is 227, and then divide by the table total at 460. So 158 times 227 divided by 460. So this expected count is 77.97. Okay, so what we really have to just concern ourselves with is we really only need to check the smallest one, because if the smallest one doesn't, if the smallest one checks, then all the other ones are going to check as well. So if we check the smallest one, I'm going to do that here. The smallest expected count is at 42. So we would have 86 times 227 divided by 460. And when we calculate this, we get 42.4. So since the smallest is at least 5, oh, whoops, I'm so sorry. So the smallest expected count at 42 turns out to be 42.4. Let me just verify that. Yeah, 42.4. So since the smallest one is at least 5, then we are okay to proceed with a chi-squared distribution. And this would be, let's say this instead of that distribution, we'll say chi-squared test for independence. We want to name it by its name or its formula. So chi-squared is sum O minus E squared over E. So observed minus expected. So the first cell was 85 minus 77.97 squared all over 77.97 plus dot dot dot. At this point, I would say go to your calculator. So when I go to do stat tests, I scroll down and I see the Goff test, but I also then see chi-squared. I want to choose C, the chi-squared test. And when I hit enter here, I get this, which is a matrix. So observed values in a matrix and expected values in a matrix. So I need to enter my data into matrix A. So to do that, so you go to second matrix, and I'm going to go over to edit. And so matrix A is the observed values. So matrix A has 3 rows and 2 columns. So it's a 3 by 2 matrix. So I'm going to enter my data, and it goes horizontally and then skips down to the next row. All right, so I enter all of my data into the matrix. And that's it. So then I'm going to quit out. And then I'm going to go back to stat tests. And the really awesome thing is that you only need to enter the observed matrix. The Goff test, you have to enter the observed list and the expected list. But for the chi-squared hop test and independence test, you don't. So you just have to enter an A, and it's going to actually calculate the expecteds for you. So I'm going to calculate here, and I get a chi-squared value of, I'm not used to working from home here, 2.065. We've got 2 degrees of freedom because we had 3 rows minus 1, and then we had 2 rows minus 1, so 2 degrees of freedom. You don't have to show this work, but I want to show you where it's coming from. And then the p-value is 0.356. Okay, now if you got lost, the calculator, let's see, calculator instructions on page 706 of your book. Page 706 talks about how to use your calculator to do this work that we just did. And just to verify, just to show you here, I said that the expected counts were going to end up in matrix B. If you go over to matrix B and you look, you can see here's the first expected count. And this is kind of nice to verify after, like I always like to look at matrix B after I've done my test, so I can just verify that I actually was okay with my expected counts. But you can see clearly that the smallest value is the one that's most important to check. Okay, and so then your conclusion is going to be like we always do. So we've got a really large p-value, right? So since our p-value 0.356 is greater than our alpha at 0.05, we fail to reject the null hypothesis. So that means we do not have convincing evidence of an association between gender and finger length. Part C, if your conclusion was an error, what type of error did you commit? Okay, well, fail to reject is a type 2. So when you fail to reject a false null, oops, that's a type 2 error. Okay, so we'll do one more full test, and I'll just go kind of quick like I did on this one too, because you can always stop and you can start the video whenever. So for this next one, so this is an actual AP question, and what's tricky about this one is I noticed that we've got a two-way table, but then I also noticed it says an association, but this is actually a test for homogeneity, even though it says an association, because of the way that the data was gathered. So take a minute, you can pause the video and read through the prompt here, and come back when you have read through it. So for this experiment, You've got 75 children randomly assigned to one of 3 groups. So because we have 3 separate groups, this is a test for homogeneity because I've got separate groups randomly assigned into those groups. Uh, so because we've got 3 randomly assigned groups in a randomized experiment, this is a test for homogeneity of populations, even though it says an association. So I understand that that can be confusing. That's why I'm talking about this problem right now. Okay, so this means that we're looking at a distribution. So all right, so we'd say something like this: the distributions of snack choice based on ad type is the same for all groups in this experiment. Oh, maybe I don't want to say that for all, because I don't want. I'm trying to make these hypotheses about something larger. So I want to be able to apply it to some larger group. So all groups of children exposed to these ads. The alternative is that these distributions are not the same. All right, so this is a chi-squared homogeneity test. Okay, if you can't spell homogeneity, it's okay. You could just call it a chi-squared HOP test. H-O-P, HOP. Okay, so we are told that children were randomly assigned to the 3 treatment groups. There's no 10% condition because it's an experiment. We're not sampling. And so then we want to look at the expected counts. All right, so the one of most concern is the smallest one. And the smallest one is this one here. So the row total is 25 and the column total is 19. And well, we have to know the table total. This is 25, 25. Let's see, 2, 7. Nope, I can't add. 4, 5, 6, 56. Okay, so for that one expected cell, large counts. So the smallest cell value is 3. And so the expected count would be the row total times the column total times the table total. So 25 times 19 divided by 75 gives us 6.33. And this is greater than 5. And so then we are okay to proceed. All right, so the cool thing is that you have your calculator at your hand, so you can actually kind of work backwards and let the calculator do all the work for you. So I'm going to go here to stat oops, actually, before I do that, I'm going to go to the matrix because I know I need to enter my data in a matrix. This is again a 3x2. So 21, 4, 13, 12, 22, and 3. And so then I'm going to go to my test, chi-squared test. I'm going to just calculate it, and I get 10 point, I'm going to write it over here, 10.29 with degrees of freedom equal to 2 and a p-value equal to 0.0058. Okay, I left this space because I like to show a couple of cells. And so I'm going to go back to matrix B where it calculated the expected values. And so then that makes it easier to do this part. So the first cell was 21 minus 18.667 because that was the expected value divided by 18.667 plus dot, dot, dot. And then I can go to the last one, which was our smallest one, 3 minus 6.33 quantity squared over 6.33. Okay, so now it looks like I know what I'm doing. All right, and so then this is very small, so we're going to reject the null. All right, so since the p-value 0.0058 is less than alpha value of 0.05, I didn't say, but you know, it's an appropriate alpha value, then we reject the null. So we have convincing evidence that the distributions of add preference, snack preference, these children would pick or differ based on add shown or add type. Okay, so I just want to breeze through a couple other little things, and you can do the reading tonight, and that will walk you through everything too. And I'll scan my notes so you can look at my full and complete notes. All right, so on the next page, this is actually in your book as well. I just copied it out of the book. It's on page 717. And I just bring this problem up because it gives you some Minitab output. And I just wanted to talk about what that tab output looks like and why. So you actually have a two-way table here. You've got males and females, and then you have, notice, like 3 pieces of information inside the table. Sorry, that's really faint. Anyway, you can look at the book and you can look at your notes, but there's 3 pieces of information here, and they're from the table here. So the first one is the observed value. This is your observed value. The next one is the expected value. So row total, column total divided by the table total. And then the third column in this output is the actual contribution of that cell to the chi-squared test statistic. So if it asks you like to do a follow-up analysis like you saw on last night's homework and 11.1 notes, a follow-up analysis is like this question here, which cell contributes most to the chi-squared test statistic. In this case, you can see this one here, this 1.883 contributes most. Part A asks you why you would use an association and not a homogeneity test, and it's all about how the data was gathered. This is asking a sample of college students. And so with a single sample, this is a test for association and independence. So you could read the rest of that. It also talks about, on page 719, the fact that you can, instead of a chi-squared, you can, chi-squared homogeneity of populations, you can do a 2-prop Z test instead. So, for example, in this male and female problem, let's just assume for a minute that, so suppose that this example above were 2 random samples. We could say that the null hypothesis is P of male is equal to P of female, the proportion of males equal to the proportion of females. The alternative would be, in this case, that it's not. It was asking about, is there a difference in fright levels? So if you read on page 719, it talks about that. I bring it up because typically on the AP exam, if you have a chi-squared, you could do a 2-prop Z test, but I'm breezing over this because chi-squared isn't going to be on your AP exam. The last thing is on page 720. What if the expected cell counts aren't all at least 8? So in this problem here, this first cell gives an expected value. This is 52 times 47 over 626, an expected value of 3.9. And so it talks about what to do if you have cell counts that don't meet the criteria. And what you can do is you can combine categories together. So this is about people who get hepatitis and if they have tattoos. And then also notice that where they got their tattoos, either in a tattoo parlor or somewhere else. So you can get them, I don't know, like I got one in my friend's living room when he was an apprentice. Some people get their tattoos in jail. I didn't go to jail. But so the question might be like, does it matter does it matter where you got your tattoo, or does it just matter that you have a tattoo? So maybe you could just collapse, these two together into one category, having a tattoo versus none. And that could be a way that you could combine them together. If you combine them together, so you have tattoo versus none, hep C or not, then this cell becomes 25. And so then now, if you do the expected values, you get an expected value of 8.48. So in practice, that's one of the things that you can do if you have expected cell counts that aren't met. Okie dokie. So that's it for tonight. I miss you, and I hope to see you again soon. If you have any questions, please feel free to message me. All right, I'll talk to you later.